You think eating a burger is just calories entering your body, nothing more dramatic. 2 hours later, your bloodstream tells a very different and unsettling biological story. Normally clear red plasma turns cloudy, thick, and pale, resembling diluted milk under microscopy. This phenomenon is called postprandial lipemia, and it signals fat flooding circulation aggressively. Your blood is no longer a clean transport system, but a congested metabolic highway. Yet, fat overload is not the most dangerous event unfolding during these first hours. Something more insidious slips quietly through the gut barrier and enters your bloodstream. This process is known as metabolic endotoxemia, and it begins shockingly fast. Fast food combines saturated fats with refined sugars, in a way evolution never prepared for. This combination increases intestinal permeability, weakening the tight junctions protecting your internal environment. As the gut barrier loosens, bacterial fragments called lipopolysaccharides leak into circulation. LPS originates from gram-negative bacteria, normally confined safely inside your intestines. Once in blood, your immune system treats LPS as evidence of active infection. White blood cells respond immediately, releasing inflammatory cytokines into tissues system-wide. This creates acute systemic inflammation, even though no pathogen is actually attacking you. Research from institutions like the NIH has confirmed this inflammatory response after high-fat meals. Importantly, this reaction occurs before weight gain, insulin resistance, or visible metabolic damage. You feel normal, yet your immune system is already mobilized unnecessarily. Inflammation raises oxidative stress, damages endothelial cells, and disrupts vascular signaling rapidly. Your arteries experience friction and dysfunction long before cholesterol numbers ever change. This is why fast food harm is invisible during the first day. No pain, no warning, no feedback strong enough to change behavior immediately. You might wonder why evolution allows food to trigger immune reactions so violently. The answer is simple. Such food combinations never existed during human biological development. Your immune system interprets this chemical chaos as danger, not nourishment. Before we go further, pause and reflect honestly on your own recent choices. When was the last time you ate fast food without a second thought? Was it within 24 hours? Or sometime during the past week? Type 24H or 1 week in the comments and answer without judgment. Awareness is the first step, because this storm begins long before symptoms appear. In the next section, the story turns darker as your brain itself becomes compromised. Because inflammation does not stay in the blood, it travels upward with consequences. By day 3, control slips, cravings intensify, and choice quietly disappears. That is not weakness, but neurobiology responding to engineered food stimuli. Let's follow that signal next, into the brain's reward circuits and memory centers. By the 3rd day, disgust fades, yet cravings intensify with strange urgency and emotional familiarity. You feel sluggish and unfocused, but one fried bite abruptly sharpens mood, attention, and motivation. This sensation is not hunger, but a neurological response to engineered food reward systems. Fast food is mathematically designed around the bliss point, balancing sugar, salt, and fat precisely. This combination overstimulates the nucleus accumbens, the brain structure governing motivation, pleasure, and reinforcement. Dopamine surges excessively, teaching your brain that relief and reward arrive together quickly, consistently. Repeated exposure dulls baseline dopamine sensitivity, requiring stronger stimulation to achieve the same effect. This is how compulsion quietly replaces preference without announcing itself to conscious awareness fully. However, reward circuitry damage is only half of the neurological disruption occurring beneath awareness. A controlled study from Macquarie University demonstrated hippocampal impairment after only 1 week of Western-style eating. The hippocampus integrates memory with appetite control, helping you recognize fullness and contextual safety. Inflammation disrupts this function, weakening satiety, signaling and impairing behavioral inhibition processes significantly overall. As hippocampal control fades, the prefrontal cortex loses authority over impulsive decision-making processes rapidly. Simultaneously, insulin resistance inside neurons reduces efficient glucose delivery to critical brain regions during stress. The brain becomes metabolically underpowered, yet aggressively driven to seek quick dopamine relief signals. This creates a hierarchy shift, where reward overrides judgment, planning, and long-term consequence evaluation. You continue eating not from hunger, but to escape discomfort generated by withdrawal-like neurochemistry. Many people misinterpret this loss of control as personal weakness or moral failure, rather. In reality, the brain is adapting predictably to repeated inflammatory and hyper-rewarding stimuli exposure. This neurological state develops within days, not decades, making fast food uniquely destabilizing to behavior. Understanding this mechanism matters because shame delays correction, while insight accelerates recovery processes dramatically. You may wonder whether stopping reverses this damage or leaves permanent neurological scars behind. The answer is hopeful, but recovery requires removing the stimulus and restoring metabolic stability. Before recovery begins, however, we must follow the damage into the liver next carefully. The liver absorbs this metabolic chaos silently, converting excess sugar into fat relentlessly inside. Unlike the brain, it offers no cravings or pleasure signals to warn you early. In the next section, you will hear its quiet distress before visible symptoms emerge. Stay with me, because this is where temporary indulgence becomes lasting metabolic injury process. By day 4, fatigue appears, digestion slows, and mornings feel heavier without obvious explanation. This is not weakness. It is your liver signaling overload through subtle systemic consequences. The liver is your metabolic command center, processing nutrients, toxins, and energy priorities continuously. Fast food overwhelms this system through volume, speed, and biochemical mismatch with human physiology. Sugary drinks deliver fructose directly to the liver, bypassing normal appetite regulation pathways entirely. Unlike glucose, fructose cannot be used by muscles and must be processed hepatically. Faced with excess fructose, the liver initiates de novo lipogenesis, converting sugar into fat. This process was designed for rare fruit abundance, not daily liquid sugar exposure. Fat accumulates inside liver cells, impairing insulin signaling and mitochondrial efficiency simultaneously. At the same time, lipopolysaccharides from the gut continue arriving through portal circulation. These endotoxins activate Kupffer cells, the liver's resident immune defenders. Kupffer cell activation triggers inflammatory cascades, creating low-grade hepatitis without pain symptoms. You do not feel liver inflammation directly because the organ lacks pain receptors internally. Instead, you feel generalized fatigue, brain fog, and post-meal heaviness increasing gradually. Blood tests may show subtle ALT elevation, often dismissed as clinically insignificant initially. Yet these changes represent the earliest phase of non-alcoholic fatty liver disease development. Importantly, this progression can begin within days, not years, of metabolic overload. The liver becomes insulin-resistant first, long before muscles or fat tissue follow. As hepatic insulin resistance develops, glucose output increases despite already elevated blood sugar. This forces the pancreas to release more insulin, amplifying systemic hyperinsulinemia further. Fat trapped in the liver spills outward as very low-density lipoproteins into circulation. These particles worsen postprandial lipemia, feeding back into vascular inflammation cycles. Thus, liver stress does not remain local. It amplifies whole-body metabolic dysfunction. You may not gain visible weight yet, but internal fat distribution is shifting dangerously. Visceral fat begins accumulating around organs, altering hormonal signaling and inflammatory tone. This process occurs quietly, without appetite changes or obvious warning signs. By the end of 7 days, the liver is working defensively, not optimally. It prioritizes survival under chemical assault rather than metabolic flexibility and balance. This sets the stage for the next failure point: insulin signaling collapse. Because insulin now faces resistance at its primary control hub. In the next section, we examine how insulin loses authority system-wide rapidly. You will see why energy abundance paradoxically produces cellular starvation and exhaustion. Stay attentive, because this mechanism explains food comas, cravings, and sudden fatigue patterns. The story is tightening, and the metabolic doors are beginning to jam. After fast food meals, crushing sleepiness appears, signaling blood sugar collapse rather than healthy postprandial rest. This sensation reflects acute hyperinsulinemia, where the pancreas floods circulation with insulin repeatedly after meals. Insulin's job is opening cellular doors, allowing glucose entry for energy production inside tissues. But constant sugar exposure forces insulin secretion all day, removing periods of metabolic relief. Cells bathed in fatty acids begin defending themselves by reducing insulin receptor responsiveness gradually. This process, called receptor downregulation, prevents glucose entry despite abundant circulating fuel in bloodstream. Muscle and liver cells close GLUT4 gates, leaving glucose stranded outside during meals periods. Blood sugar spikes higher, demanding even more insulin to force compliance from pancreas cells. This creates a vicious loop of rising insulin and worsening cellular resistance throughout body. Paradoxically, energy is everywhere, yet cells experience functional starvation internally during abundant states repeatedly. Fat burning shuts down because insulin blocks lipolysis aggressively in tissues across the body. Meanwhile, glucose cannot enter cells efficiently, deepening fatigue and brain fog after meals periods. This explains food comas, irritability, and the urge for more quick carbohydrates soon afterward. Insulin resistance here is acute, not permanent, but it escalates rapidly with repetition daily. 7 days is enough to reprogram insulin signaling expectations system-wide in humans very significantly. This temporary state sets conditions for chronic disease if patterns continue unchecked over time. But the story is not hopeless, because insulin sensitivity can return quickly with intervention. Recovery begins when inflammatory inputs stop and cellular signaling regains clarity again soon afterward. However, one system suffers collateral damage that insulin alone cannot explain by itself fully. Your gut microbiome changes dramatically, reshaping metabolism, immunity, and future weight regulation patterns quickly. This shift may be the most consequential outcome of 7 fast food days for health. Because microbes influence inflammation, insulin response, and appetite signaling continuously in humans over time. When they change, your entire metabolic future tilts quietly without awareness at first period. In the next section, we meet the microbiome and its protective collapse during fast food. This is where short-term choices create long-term metabolic inertia and risk for disease development. Stay focused, because repair is possible once you understand what was lost here precisely. The greatest damage after 7 days does not appear on your waistline, but inside your gut. Your colon hosts trillions of microbes that regulate immunity, metabolism, and inflammatory balance daily. These microbes require dietary fiber to survive, communicate, and maintain intestinal barrier integrity. Fast food contains almost no fermentable fiber, effectively starving beneficial bacterial populations rapidly. As fiber disappears, protective species like bifidobacteria and Akkermansia decline sharply. These microbes normally reinforce the mucin layer lining your intestinal wall. Without nourishment, they retreat, thinning the mucus barrier separating bacteria from intestinal cells. Opportunistic microbes then begin consuming this mucus layer as an emergency food source. This erosion further increases gut permeability, accelerating endotoxin leakage into bloodstream circulation, At the same time, sugar and fat-loving bacteria expand aggressively in this altered environment. Many belong to the firmicutes group, known for extracting excess calories efficiently from food. This microbial shift increases caloric harvest, even when total intake remains unchanged afterward. It also amplifies inflammatory signaling, worsening insulin resistance and fat storage tendencies system-wide. Researchers have shown this imbalance predicts future weight gain, independent of calorie intake. This is how short-term fast food creates long-term metabolic momentum quietly. Your microbiome begins training your body to expect, crave, and store energy differently. Signals sent from gut bacteria influence hunger hormones like ghrelin and peptide YY. Cravings intensify not from willpower failure, but from altered microbial messaging loops. The gut-brain axis becomes biased toward energy-seeking rather than regulation or restraint. Importantly, this microbial damage occurs rapidly, but it is not permanent if addressed. The microbiome is resilient, but only when given correct substrates and recovery time. Ignoring this shift allows inflammation and insulin resistance to persist beyond dietary correction. This explains why many struggle to rebound after short junk food periods unexpectedly. They fixed calories, but forgot to restore the ecosystem controlling metabolism underneath. Before ending this story, one critical question remains unanswered. How long does the body need to repair this damage once fast food stops? Is recovery measured in days, weeks, or months of disciplined correction? The answer is surprisingly hopeful, but it requires deliberate strategy and restraint. In the final section, we outline how repair actually unfolds safely and effectively, because your body is not fragile, but it demands respect after insult. The good news is simple: 7 days of damage do not equal permanent metabolic destruction. Your body is adaptive, resilient, and designed to recover when harmful signals finally stop. The liver leads this recovery because it alone possesses remarkable regenerative capacity among organs. Once sugar and refined carbohydrates are removed, hepatic fat oxidation resumes almost immediately. Within days, liver glycogen depletes, forcing stored fat to exit hepatocytes gradually. As liver fat decreases, insulin sensitivity improves system-wide without heroic interventions required. This alone reduces insulin demand, inflammation, and post-meal fatigue dramatically for most people. However, liver recovery depends on simultaneously calming the gut-derived inflammatory stream. Restoring the microbiome becomes the next critical repair priority after fast food cessation. Soluble fibers act as prebiotics, selectively feeding beneficial bacteria starved during the experiment. Foods like legumes, oats, vegetables, and berries rebuild microbial diversity steadily. Fermented foods introduce live cultures that accelerate mucosal healing and immune recalibration. As the mucus layer thickens, endotoxin leakage declines, and systemic inflammation recedes. This reduces immune activation, cortisol signaling, and stress-driven metabolic dysregulation gradually. Brain clarity often returns before body weight changes, signaling reduced neuroinflammation first. Cravings weaken as dopamine sensitivity normalizes, and gut-brain signaling stabilizes again. Importantly, recovery speed depends on prior metabolic health and accumulated insulin resistance. Individuals with existing fatty liver or type 2 diabetes require longer, cautious recovery periods. For them, 7 fast food days may trigger acute flare-ups needing medical oversight. This is not fear-mongering. It is respect for biological thresholds and individual variability. One fast food meal does not destroy health, but repetition trains the system negatively. 7 consecutive days represent a meaningful metabolic challenge, not a harmless indulgence experiment. The lesson is not perfection, but awareness of cumulative biological consequences. Every food choice sends instructions to insulin, the liver, the brain, and microbes. Over time, these instructions shape resilience or fragility without moral judgment involved. When standing at a counter, ordering convenience, remember the week you just witnessed internally. Not guilt, but memory should guide your next decision consciously and calmly. Your body is not punishing you. It is responding intelligently to signals provided. Change the signals, and recovery follows with remarkable efficiency and speed. If this explanation shifted your perspective, share your experience in the comments below. Like the video if understanding biology helps you make calmer, wiser health decisions. This knowledge empowers choice, and choice is where long-term health quietly begins.